so our topic today is mitosis. And let's begin with our goal. Okay, by the end of this video, you will be able to explain the stages in mitosis. Okay, it's a quick review. Now, let's begin with this question. Why do cells have to divide? The simple answer is, number one, they have to grow and develop, so they have to divide. Number two is to make the sex cells, the sperm and the egg, and lastly, to replace the damaged cells. Now, for unicellular organisms like bacteria, the cell will divide in a very simple and fast way. The cell grows and gets bigger. After some time, it will copy its DNA, and the cytoplasm will divide into two new organisms with two DNAs. This is called binary fission. In multicellular organisms like humans, there are two ways that cells divide, and that is mitosis and meiosis. We're going to take a look at mitosis. But let's start with the two types of cells in our bodies. We have body cells and gamete cells. The body cells have two sets of chromosomes, and we call it as a diploid cell. It has two sets of chromosomes, and they divide by mitosis. However, the gamete cells have only one set of chromosomes, and they are formed by meiosis. It is also called a haploid cell because it has only one set of chromosomes. Now, we refer to the number of chromosomes in the gametes, the egg and the sperm, the number of chromosomes as N. So in human beings, N is equal to 23. And we refer to the number of chromosomes in the body cells as 2N, okay, because it has double the amount of chromosomes in the gamete cell. So this is the stages of mitosis. Mitosis is when we divide the nucleus. And after mitosis, we're going to divide the cytoplasm, which we call cytokinesis. Let's take a look at the different stages of mitosis. Before mitosis, we have interphase, which is not part of mitosis. Okay? In interphase, we have three stages in interphase: the G1, the S, and the G2 phase. What happens in the G1 phase? The cell is in a normal activity, it's doing normal activity, like it is making enzymes. It's just growing and developing, okay, getting bigger. And it's also going to double the organelles inside the cell. After G1, the cell will go to the S phase, and at S phase we're going to copy the DNA because we're going to have two new cells, so you have to copy the DNA and share to the cells. After the S phase, or the synthesis phase, you're going to have the G2 phase, and this picture shows the G2 phase. In G2 phase, we can see that the DNA has been copied or duplicated, and there is nuclear loss inside the nucleus. There's also the nuclear membrane. Now the DNA looks like a bunch of hair, and we call this DNA as chromatin. Inside the cytoplasm, we have a structure we call the centrosome, which is made up of a line-shaped microtubules or spindle fiber, and this structure, the yellow ones, called the centrioles. This is interphase. Okay. Now after interphase, mitosis can begin. We start with prophase. In prophase, we can see a few changes. The first one is we can see that the chromatin, the DNA, is starting to be thicker, and we call that chromosome. Okay, when it looks like this, we call it chromosome. We can also see that the nucleolus has disappeared, and the centrosome are beginning to move away from each other. This is prophase. If you look at the chromosome in this picture, you can see it has two arms, and they have a center. They are joined together at the center. We call this center centromere. Okay, joined together at the centromere. And we call each arm as sister chromatids. Okay, so two sister chromatids joined together. After prophase, we have prometaphase. And we can also see some changes in prometaphase. The first change is that the nuclear membrane has broken down and disappeared, and the spindle fiber has attached to the centromere of the sister chromatids, and also the centrosomes have moved to opposite poles of the cell. The next phase is metaphase, and in metaphase you can see that the sister chromatids or the chromosomes are aligned in a straight line. We call this imaginary line as metaphase plate. And finally we have anaphase, where the spindle fiber will begin to pull the sister chromatids apart and break them at their centromere. Now they are attached, the spindle fiber is attached to the centromere, where there is a protein which will help to separate the sister chromatids. We call the protein as kinetochore. After anaphase, we have telophase, and in telophase we can see some big changes. You can see the nuclear membrane has started to form, and the chromosomes have become thinner to form chromatin. The nucleolus has also appeared, and this is telophase. After telophase, mitosis has finished, and we're going to divide the cell, the cytoplasm, which is called cytokinesis. Cytokinesis happens differently in animal and plant cells, so let's take a look. In the animal cell, the cell membrane is going to fold in by a help of some tubes or some tubules, proteins we call actin. Okay, they form the actin ring, and they fold in and meet at the center. They form something we call a cleavage furrow. In the plant cell, because of the cell wall, we cannot fold it in, so we're going to create a cell plate. These are just small cell walls which will grow and connect to each other, and finally join together to form the new cell wall. Now the cell division or mitosis is not random process. It is very controlled, and it's controlled by some proteins we call cyclins. They control the cell through checkpoints, and the checkpoint is just a normal, it looks like a normal police checkpoint on the road. Okay, and in the checkpoints on the road, when you drive in and you meet the police, they will stop you and check if everything is okay with you, with your car, and they will let you go. This is similar to how the cell works and controls mitosis. So let's take a look at it. The first checkpoint is in G1. Before the cell can go from G1 to S phase, we have to check for nutrients. and check if the cell is growing, and lastly, if there is any damage in the DNA. If there is any damage in the DNA, the cell will try to fix it before it will move to the S phase. If the cell cannot fix it, it's going to receive signals from inside the cell or outside the cell to kill itself, which we call apoptosis or cell death. If everything is fine, we move to S phase and copy the DNA, and we move to the G2 phase. Before we can start mitosis, at G2 phase we have to check again. Check the size of the cell and check if the DNA that we copied is okay. If there is a problem with the DNA we copied, we have to fix it before we can start mitosis. And finally, we have a checkpoint at the metaphase phase, at metaphase. That is, we're going to check if the spindle fiber is attached to the chromatids. We check very well to see that they are all attached, so that they can separate into the new cells. So this is how the cell controls the mitosis. Okay. Now, what could happen if the checkpoints do not work? Would that be a problem? And the answer is yes. In a normal cell, again, it is very controlled. If the cell cannot control the mitosis, the way it divides, it's going to form cancer cells, which will just divide many times, to divide and cause cancer. Okay, and it's a very serious problem around the world. Many people die from it every year. Now, let's talk about the parent cell. We started with one cell we call parent cell, and we divided it and have two new cells we call daughter cells. If you compare them, what is the difference between the parent cell and the daughter cell? And clearly there is no difference. They are exactly the same. Both of them are diploid and they are exactly the same. Okay, so thank you for watching. I hope this is helpful. See you again next time.